I'm really excited to be here and give this talk, and thank you all for coming to something that's a bit far afield from what maybe many of you think about usually during the day. So thanks for humoring me and in letting me tell you a little bit about what we think about in neuroscience and some circuits in the brain that underlie navigation and other cognitive behaviors. So this is the talk. I think I changed the title, but it doesn't really matter. The same content that I intended to give, which is I want to give you an overview of the recent discoveries in neural representations in the brain for abstract cognitive variables, and the method for discovering these representations, and also trying to give you some idea of how these representations fit in with the computations that the brain performs in generating rich behavior. So, so let's start with the behavior and the computations. And so I love this example. This is Clark's Nutcracker. It's actually one of many food caching birds, many jays that cache their food. And they have this remarkable behavior, this remarkable ability for spatial memory and recognition. So let me tell you a little bit about what they do. So in the fall, they start to cache food for the winter. Okay, and so that it involves, finding pine seeds and then burying them in the ground. And typically in the fall, they'll do something like cache food in 2,000 locations, about 10,000 seeds in all, about 100 sites per hour at the peak. Okay, for, at the peak, caching time. And typically those cache sites are the bases of various trees, but the thing is that those trees are widely distributed, 20-mile range or so. And they do, with decent and relatively high fidelity, retrieve food from those caches. So over a 20-mile range, it can't be a random search over all the bases of the trees. And moreover, it's an interesting problem because they then go back and retrieve these caches in the winter, when the ground looks quite different than it did in the fall when they first cached the seeds because of snow and so on. And this is it is a biological memory process because towards the end of the winter and over time the memory does fade of food caches and the retrieval accuracy goes down. Here's quite a different example of complex spatial behavior in animals. This is the burrow system of the kangaroo rat. It's this one-dimensional underground burrow. It's a one-dimensional burrow, but itself, each passageway is one-dimensional, but it forms this three-dimensional mass under the ground. And there are many different entrances and exits, I think marked by Z's. And so, really the animal can, enter from these various entrances and then presumably exit from the various exits. And when it's in the burrow, even though it's dark inside and locally looks very similar everywhere in the burrow, and there are many T-junctions, et cetera, it somehow presumably knows how to get out and without doing a full random search each time. And so, in general, the problem of, knowing where you are in space as you move around in that space, it involves a lot of computations. It involves integration of your velocity to update your estimate of position as you move around. It involves integration itself, involves memory, because you have to remember what state the integrator was in before incrementing any change in the representation. It involves a lot of inference, like in the food caching bird example. In this example, there's a lot of ambiguity in the spatial data, right? There's many distinct sites that look the same. The same site can look different from day to day, and so there's a lot of this aspects of pretty non-trivial probabilistic inference that must be performed by these circuits. The inputs are discrete and continuous in time. There's like velocity, which is like a self-motion estimate, which is continuous in time, but there are also discrete things like discrete landmarks, and these also need to be integrated into the estimate of position. In robotics, people have to deal with many of the same problems when they send out search and rescue robots, to like search disaster areas. They need to build maps of the environment and then also be able to then find their way through the environment, flag the locations of people in the environment, et cetera. So in the domain of robotics, this problem has been formalized, and it's called the problem of simultaneous localization and mapping, or SLAM. And in SLAM, this is just to illustrate some of the complexity of SLAM. So this is like an agent going through an environment, a building in this case, and it estimates its position and thereby assigns position estimates to each wall and other things in the building that it encounters, and builds a map. And if it builds a map, the map looks like this. So I don't need to tell you that this is not actually the footprint of an actual building. This is just a distortion that's induced by the really noisy estimates that we have of our position position based on integrating velocity cues because, these velocity cues are error prone, they're only approximate, and then the integral of that accumulates error over time. Right, so it's a really accumulating process. So by contrast, what the agent does and can do, if it's a fully probabilistic agent, it can make some inferences about position based on the cues it has, and then build a crude map, and then use that crude map to correct its estimates and do a bootstrapping procedure that's probabilistic, and actually build an accurate map from that combined bootstrapping procedure. Okay, so these are the kinds of problems that animals have to solve as they navigate through space. So let me just say a couple of things about the SLAM problem again. So SLAM problem is has two parts. There's the mapping part, which is what is the world around me? Like what is there? Right, and where are those things that are there, right? So you have to sense the world from various positions, integrate measurements of where those positions are to build a map, and but you can do this if you have perfect knowledge of position, right? So if you can walk to each object in the room and you know exactly what your coordinate is, then you have built a perfect map in one shot. Right. On the other hand, there's the second problem of localization, which is suppose you have a map of the world. I know where every object is in the world, and I just need to know where I am within that map. So then you can sense the world. You can relate your sensor readings to the map of the world, and you can now compute your location relative to these different objects in that map by triangulation, and you've got perfect localization if you've got a perfect world model, right? So each of these problems on its own is pretty solvable. It's the combination, that simultaneous combination of those two that makes it non-trivial, right? So given imperfect versions of each, it's hard. Okay, so now I want to also point out that the SLAM problem is actually generalizes past the domain of spatial localization and spatial navigation, even though it's been formalized in that domain. It's really the combined problem of what and where, right? And anytime you have a combined problem of what and where, you're sensing what from various different wheres, then you can think of that as a SLAM problem. So for example, if you want to do visual inference or 3D reconstruction of a 3D object from various different views, you can think of that as a SLAM problem because you need to know from where you're looking at that object and what that 2D view is, and then you can construct a 3D object. Similarly, like in microscopy, for example, if you're doing microscopy of a wet lab sample, a protein that's moving around, or it's moving around relative to the camera, then there's again the question of from where am I looking at the molecule and what is the structure of the molecule from that angle. And if you had both those pieces of information, you could immediately reconstruct it, but then without accurate information on the position of the molecule or the structure of the molecule, you have to bootstrap simultaneously. Okay, so that's the SLAM problem. So what are the substrates in the brain for solving some of these these SLAM problems, right? Navigation problems. Unfortunately, we have worked on SLAM. I'm not going to be able to tell you too much about the probabilistic inference part of it, but I want to tell you something about the different players in the brain that are contributing to the computation. Okay, so here's the brain. Here's the human brain. And here within the brain is a structure called the hippocampus. Hippocampus is famously known to be involved in memory. This is known from human studies in patients that had damage to the hippocampus, the most famous being patient HM, who, when his hippocampus was damaged, had a complete inability to remember new events in life. So he was not able to remember, 10 minutes after meeting somebody that he met somebody and where he met that person, etc. And but he had really good memory of things, far in the past. He continued to have really good memory of completely different types of things, like how to ride a bicycle, so his motor memory was unimpaired. Also some semantic memory, just knowledge of basic facts, who was the president during World War II, etc., were, I think, relatively unimpaired. So, and linguistic abilities were spared. So it's a very particular form of memory in the hippocampus. It's a conjunction of like who, what, when, and where events, or biographical events that happen in life. Okay, so that's the hippocampus, and the reason it's called the hippocampus is that, when you actually look at this physical structure, it has that shape, which looks a lot like a seahorse, whose name in Latin is hippocampus. Okay, and so the hippocampus itself has been, beautifully characterized. There's You can see all of these substructures in the hippocampus, which consists of dentate gyrus, CA3. You don't need to know these names. I'm not going to CA2. And then it'll show us CA1, which is another important subarea of the hippocampus. And this is in rodent brain. And the subiculum, which is sort of the output of the hippocampus back to the cortex. So, so there's all this like beautiful richness of structure in the hippocampus is a laminar structure, organization of cells, in these laminar structures and projections between these different substructures. So there's a lot of complexity in the different hippocampal subcircuits. And in fact, just to situate you in where the hippocampus lies in a connectivity sense in the brain, this is a very this is a famous diagram of the visual system by Van Essen and colleagues, and it's really used for like the visual processing, in the visual processing context because it shows the visual stream. Here's retinal ganglion cells in the retina, the thalamus that gates information to cortex, low-level cortical areas, all the way up. And then I just want to point out that at the very top, there are these two areas called ER, which stands for entorhinal cortex, which is a high-level association cortex that receives multimodal, multisensory input from all the sensory hierarchies. So basically, you can think about this as one slice from the visual side projecting to entorhinal cortex, but then there's a corresponding auditory, somatosensory, and other cortices that also have this hierarchy and then funnel into the entorhinal cortex. Entorhinal cortex then is the main gateway from cortex into this structure, the hippocampus, which is at the very top HC of the hierarchy over here. Okay, and so we're going to focus on, I don't want to overwhelm, that was just to show you where it sits, so we're not going to be concerned with all of those areas in detail, but I wanted to show you some sub-circuit that we are going to be thinking about. So this is entorhinal cortex, this area that funnels all cortical information to the hippocampus. This is the thalamus, which is used for controlling, routing, and gating the flow of information into the brain, into the central nervous system, the cortex. So that sends input to the entorhinal cortex. Entorhinal cortex projects to these various subfields of the hippocampus, and CA3 and CA1 in particular. And we're going to be talking about head direction cells, grid cells, and place cells in these three areas respectively. Okay, so I've told you a lot about the complexities of the behavior in animals, right? As they navigate in the world, and I've also, shown you something about the complexity, structural complexity of these different neural circuits. And the question is, amidst all this complexity, can we glimpse something about, something simple? Can we glimpse some of the simple computational representations that the brain uses to solve these problems that we've talked about? Or, in other words, maybe to make a bit of a stretch, if we think about the mind as, the extraction of key latent variables in the world that are simple variables but not provided directly by the world to us. So we think about the mind as extraction of these like essential important variables from the world, then can we glimpse something of the mind from the brain? That's the question. Okay, so the answer, the brief answer is, I think we can. And the statement, my assertion that I want to make and then support in the rest of the talk is that the brain contains strikingly low-dimensional and rigid circuits for symbolic representation of key latent variables. And at the end, I'll show you how it also contains some pretty non-rigid structures that allow for generalization of those.
So I suspect you're going to tell us what all of this means, but I'm particularly interested in rigid circuits. It's not obvious to me.
What that means? Yes, and I will explain that. And by rigidity, what I mean is this is, the question is, okay, there's many ways that we can define rigidity. We can ask about, the states that or the dynamics of a circuit over the lifetime of an animal, right? We can ask about that. We could ask about the dynamics of the circuit across different tasks as the animal performs those different tasks or different behaviors. And the question is, are there circuits that do the same essential computation across tasks and behaviors? And then are those responses conserved over the lifetime of the individual, or are they constantly changing? And what I want to say is I first want to say that there's actual constancy and variance over task and over lifetime, and that's what I call rigidity. But at the same time, the animal has, the ability to represent new tasks and do that in a reconfigurable way. But these are done by downstream circuits that use some of these rigid representations, but also have flexibility built into them. So yeah, it's a great question. Hopefully, I can clarify that as we go along. Yeah.
Is consistency across individuals also part of that?
Yes, absolutely. Absolutely, that's right. So it turns out that if one sees certain really conserved circuits within an individual, then that circuit tends to be highly conserved across individuals of the same species as well. Not only individuals of the same species, but across a lot of species. So it's quite interesting, and across Mammalia for many of these representations. Okay, so any other questions before the next part? Okay. So I want to tell you, I want to motivate a method and or a general principle, motivate a question in a sense. So the question is the following, right? So can we look at neural data, just spike trains, just a time series of spike trains coming off of an electrode in the brain, and somehow discover just from that spike train what those cells or that circuit is representing and what it's doing? Can we do this, or do we necessarily need to have an explicit understanding of what variables being encoded? So can we do things, by just looking at the time series and infer what's being encoded rather than know what's being encoded and then look at the encoding of it in the brain? So now, the point is that there's a lot of inputs to the brain that we don't fully characterize, right? Like even an experimental setting, you can characterize some subset, you can control some subset of the variables, but you do not have control over all the variables that the animal may be attending to, right? Or that it may care about, or that impinge on the animal's sensory system. The brain also has its internal states and internally generated dynamics, which again, you cannot control, right? So this becomes really important because if we assume some sense of control and then try to regress neural activity on these controlled variables, you're probably missing a lot, right? So the hope is that we can do something by just looking at the activity and say something about the variables. So what do people commonly do in neuroscience? The common thing to do is, like I said, you find or control some independent variable. You run an experiment which is very controlled. You control the stimuli going to the animal, or you control the animal's actions in a low-dimensional motor task or something like that. And then once you control this variable, then you characterize the neural circuit, and you characterize the neural responses by regressing the activity of those neurons onto that variable. And thereby, you've characterized how that neuron responds to those variables, right? And so it's a supervised technique. You need to know the variable that's being encoded. Plus, you also miss all the variables that you are not measuring or regressing against, right? There's no way to know which other variables that system is encoding unless you could go and list every possible variable in the world and then eliminate the possibility that those neurons are systematically representing it. And then decoding from other behavioral states, one thing that has been really beautiful and fruitful in the spatial circuit is it's possible to record from neurons, characterize what they do during some task, and then assume that the same characterization applies, that the neurons are doing pretty much the same thing on a circuit level when the animal is, say, resting, and then do the same. You just decode based on this encoding model. You decode the neural responses during rest or some other behavioral state, and you assume that the encoding model hasn't changed, and thereby decode what's going on during rest. And so that leads to the discovery of things like maybe some reimagining of past events or past activities in these neural circuits or during sleep, right? Like kind of a dreaming-like decoding. However, it depends on the assumption that the circuit is the same during these states without any method to verify whether that's true. Okay, so instead what we're going to do is we're going to assume that the independent variables are not known, or at least we're not going to use them in our approach, and we'll let the behavior be relatively unconstrained. And then the idea here is to then take the shape and geometry of the data itself. Just look at the neural data, this time series, and characterize something about it. Its shape and geometry in some response space, and then we, based on the shape of the response, we then decode some latent variables on that space. Okay, and so this approach has been inspired by a lot of theoretical work in the past, as well as some recent experimental works that applied those theoretical ideas. So today I'm going to tell you primarily about a couple of works recently, one from my group and one from the group of David Tank, done by Sam Lewallen here, who's going to give the second talk, and his collaborators. Okay, and so the third part is decoding from other states. So now, we can, without explicitly assuming that in a different behavioral state the same coding applies, we can now look for coding in other behavioral states and check whether that's true. Okay, and so our goal is to go towards a fully unsupervised or blind decoding of neural responses with no averaging or trial structure. So let me illustrate what all of this means. Okay, so let's just be a little bit more concrete. So let's look at this circuit that I showed you. Now we're going to just look at the first part, which is this thalamic area that sends information to entorhinal cortex and that is known to contain these cells called head direction cells. So these head direction cells are cells that are known to encode the direction of which way your head is facing in some room with respect to some external reference. Okay, so it's not head relative to shoulders, it's which way the body and the head are facing, the head is facing relative to the external world. So it's truly like a compass in the brain. Okay, it's really like maintaining some compass type bearing, and it does so even in the dark when you turn off the lights, etc. Okay, so this area contains these compass cells. We don't know what else these compass cells encode, and we don't know if that's really the exclusive encoding that these cells do. So what we did is we took an unbiased sample of cells recorded in this whole area by our collaborators. We take this time series, and now we bin it so that each narrow time bin is a vector of dimension n, where n are the number of neurons that are recorded in the sample. In this case, the data set contains 10 to 100 simultaneously recorded cells, typically 30. So we're looking at about 30 cells. And in fact, we have this data taken during awake exploration as the animal's running around in a 2D space, like an open field and foraging for Cheerio pellets that are thrown, also during REM sleep and non-REM sleep. And we have ground truth data available for the actual physical head direction of the animal as it's running around, but we don't use it for any of our decoding approaches. We only use it to validate the method in the end. So let me just very briefly summarize what we can do with this time series. So here again is that time series of data. We bin it. Each moment in time, the response is this n-dimensional vector, so it's a point in this n-dimensional space. And together across all time, the data forms a cloud, a point cloud in this n-dimensional space. So this is a schematic of that point cloud over here in this n-dimensional space. Okay, so now what we want to do is we want to characterize this cloud, the shape of this cloud in this n-dimensional space. So to do that, we do a couple of things. We can apply topological data analysis methods, which involves basically looking at the data at different spatial scales. So it's like taking the data point cloud and looking at various levels of blur. So just adding balls of different radii to each point and looking at blurs. So now when the data have a certain level of blur, you can see that the only sort of the connected, you characterize all the connected groups and their shapes at different scales of blur. And so this scale, there's a fully connected, like a ring that emerges with like a hole in it, and that hole persists over many different scales until at some scale it's filled in again. Okay, so then we can characterize the existence of these different shapes by these different Betty numbers. So like a Betty number 1 corresponds to a ring with a single hole in it, and then higher order Betty numbers characterize the existence of other things like tori, etc. in the data set. So we can apply these methods to characterize the topology of the full point cloud. And then you can also use local dimensionality estimation methods like correlation dimension to estimate like local dimension locally on the manifold. Okay, so now given some estimate of the topology and the dimension of the manifold, we can now fit spline, like meshes or splines, in this case, in the 1D case, to the manifold. And so we can do that. And now we can parametrize the spline with the parameters of matching dimension and topology. Right, so in the case of a 1D circular variable, it would be a parameterization with a one-dimensional circular variable alpha. Okay, so now we get a latent variable that we've extracted from the data set. Now, given any point in this point cloud, we can now just project it down to that parameterized spline, read off the value of the parameter at that point, and that gives us an unsupervised decoding of the state of this network with respect to this latent variable. Okay, so that's our decoding technique, and so here it is on the data. So this is the topological characterization of the data. There's across many scales, there's like one big fully connected component, and then there's one H1 feature in these Betty barcodes. So this is like the existence of a ring that persists over many different spatial scales, and there are no persistent higher order Betty numbers. Okay, so this is the real data, and I want to show you what that looks like. Oops. So let's have a look at the data. So this is for your viewing pleasure only, that we've projected it to a lower dimensional space. This method is, dimensionality reduction is neither necessary nor sufficient for this method. Okay, so it's not necessary because we can do the whole topological characterization in high dimensional space. It's not sufficient because if you embed the structure, and I shouldn't talk over this video because it's my favorite video in the whole talk. This is this is a circuit of thousands of neurons. But we have 30 of them, 35 of them, so it's 35-dimensional space, and you can see clearly that the data form like something that looks like a one-dimensional ring, although it's very convolved, like convoluted ring, right? And so if you project this, if you want to do dimensionality reduction and extract a latent variable, the minimum embedding of a 1D ring is 2 dimensions, right? You have a 2-dimensional embedding, which is not the real latent variable, right? The real latent variable is this 1D thing. So once you have something topologically non-trivial, then dimensionality reduction itself is not sufficient to recover the minimum latent a dimensional latent variable. Okay, so now we do the parametrization of this, for the spline to this manifold, we parametrize along the spline, and now we can plot this latent variable that we've extracted in an unsupervised way. We call it SPUD for spline parameterization for unsupervised decoding, and we actually, in blue, and then we plot the physical measured direction of the head of the animal in black. And of course, this is, there was one choice of direction of movement, right? Direction of change of alpha, and one choice of the origin, which are both arbitrary. And so we choose those, but up to that, you can see that these are an incredibly high accuracy match, yeah.
How do you infer like the velocity as you move up? I mean,
yeah, that's a fantastic question. What is? How do we choose the parametrization? How do we change the rate of? How do we change alpha as we move along the ring? So that's a choice, and it's not obvious what that choice should be. You could imagine choosing something based on the density of the data, but then that's sort of induced by our sample of the data. Instead, we decided that we're going to do an isometric parametrization, which is just to say that if you move equal distances in the along the manifold, you have equal changes in the alpha. So it's an isometric parametrization, which is actually a rather strong assumption. It's saying somehow the brain is devoting equal neural resource, or it's like some kind of real translation invariance or rotation invariance built into the coding structure. And that's actually, I think, really an important insight into how the brain is encoding this variable. It's really saying that basically every angle is equally important in its representation. And it need not have been so. So we know that in the encoding of visual stimuli, there's in visual cortex, there are neurons that represent the orientation of edges in the visual world. And there we know that the neural representations for edges that are in the cardinal directions, vertical and horizontal, are overrepresented relative to all the other directions. That is not true in this encoding.
So just to rephrase to make sure I understand.
Yeah.
A constant speed rotation in the world maps onto a constant speed traversal of this ring.
Along the manifold space. That's right, and not in Euclidean space, but in this manifold space.
In the manifold space.
That's right. That's right.
Well, but that's defined by its position in the ambient space, right?
It's in the high dimensional ambient space. That's right, but it's like the curve length.
Yeah.
It's the curve length, right? Arc length. Yes, it's the arc length. Okay, so now so we can do that decoding, and now we can also construct things like the traditional tuning curves that people construct. So you can, these are plots of the firing rate of individual neurons as a function of the latent variable alpha. And you can do the same thing and do the supervised decoding, which is the regression of the neural firing rates onto the measured head direction, theta. And you can see that in a completely unsupervised way, we can recover these supervised tuning curves, again, up to this rotation and shift. Right? So this is a pretty accurate decoding. Now, I should also say that actually the unsupervised estimate is closer to the brain's internal state, its internal estimate of head direction, rather than the externally measured head direction measured by experimenters. So I'm just showing you that it's a narrower distribution of errors between this latent estimate and a supervised decoding of brain state that's narrower. They're closer to each other than the latent is to the measured head direction theta. And why is that? Well, there's a few reasons. One reason is that actually remember that there is no external sensory cue, single cue that tells the animal what its head direction is. It's an inference based on, as I move around, I can only infer which way I'm facing. I don't have perfect information about which way I'm facing, one. Second, there can be errors in the measurement of head direction relative to the real head direction. And third, this is a cognitive circuit, and it's well known that in different spatial cognitive circuits, sometimes the animal does not represent its current value of the variable that's being represented, but rather like it thinks ahead to where it's going to go in the future or where it's been in the past. So there's many different reasons why the mental state can be different from the actual physical state, and this latent variable better tracks the mental state than the physical state. Now you can ask, well, okay, to what extent is this ring really one-dimensional? That's kind of what you want to know. How much, it's a finite thickness, and so how much information is hidden in this perhaps low variance, but still, thick dimension. And so, and this was our goal, right, to uncover any other encoding. And so what we can do is we can plot the manifold itself in blue, and then in red is basically taking synthetic data, generating synthetic data based on this latent variable alpha, and just adding point process noise to the response of single neurons with the variance, coefficient of variance per neuron that's matched to that in the data. Okay, so it's basically like saying that the thickness of the data are fully explained by just simple point process IID noise per neuron. So it doesn't look like there's much there, but we can also take the correlation matrix of neurons and mod out the latent alpha, the latent variable part of the encoding, and look at what the residual structure. Okay, sorry, here we've modded out the measured head direction. You can see there's still some residual structure in the correlation matrix. Here we've modded out the unsupervised decoded estimate, and you see that there's no structure left in the correlation matrix, suggesting that really this seems to be down to the floor, noise floor. There's no other information being encoded in this thousands of neuron circuit. So it's kind of shocking that thousands of neurons are devoted to coding this one-dimensional variable, but it seems to be true. Okay, autonomy of the representation. So here I show you that if you look at this same circuit, same neurons just measured an hour or two later when the animal is sleeping. Then during REM sleep, you can see that, again, the data look like they have some low-dimensional structure. In fact, it looks like I forgot the slide. So basically, there's a co-embedding. We can just co-embed the sleep and the wake data, and they lie right on top of each other, like right on top. Okay, so one completely overlies the other. And so, I'm sorry, you'll have to take my word for it because I didn't have that slide in there. So it seems to be that the brain is now not just generating these head direction states when awake and exploring in the two-dimensional world, but it's also autonomously generating them. So this is a circuit that has enough stability of its own to, even during sleep states when there's no sensory data arriving related to position in any sense, it's able to at least visit all the same states and only those states and nothing else. Okay, so now what can we say about the dynamics of the states in the circuit in the autonomous system? Like what is going on as the animal is traversing these states during sleep? Right? Like, is it doing something systematic? What's going on, right? So we can actually characterize that, first of all, there's no net flows into and out of particular states in the manifold. So there's not any sources or sinks along this manifold, this 1D ring. They all seem to be relatively, neutral states. And so the system, if has equal flows in different directions. But if the state is knocked off the manifold, then it tends to have large net flows back towards the ring, and we can quantify this in various ways in higher dimensions. And so it would suggest that this system is really a set of attractive states. This is an attractor. It's a manifold where the states are stable and attracting. So states off of it are attracted back to it, and along, it's like neutrally stable, okay, from an energetic sense. And we can characterize the dynamics as really being diffusive during REM sleep. So the system, we can now trace how the states evolve, starting from some state just over time during REM sleep, and this is kind of like the snaking around of the state. This is a 1 second long, the purple just encodes the last second of trajectory, and you can see it's sort of like moving around, and we can quantify it and show that the dynamics are diffusive. So if this is the true dynamics, and this is the dynamics during waking, and actually this curve of square displacement versus time is quadratic during waking, which is consistent with directed motion along the ring, because if there's a directed consistent velocity along the ring, then the squared deviations will be quadratic. On the other hand, during REM sleep, the square deviations are actually linear, which is characteristic of a diffusion process, and they're also local. So those two together characterize a diffusion process, and it turns out we can do some further analysis. We have some theoretical prediction based on models of this head direction circuit. So the finding is that actually, if the system's diffusive dynamics are driven by high-dimensional IID noise per neuron, then that amount of noise, because it's IID and averaged across all the neurons, will not contribute to this amount of diffusivity. So it suggests that actually the noise coming in to drive, or whatever's driving the system during sleep, is actually coming in through a specific low-dimensional input that's aligned to the dimension of the manifold, so that it can, in an efficient way, push the state around the manifold, which suggests that really it is an input coming in aligned to some maybe angular velocity input that normally during waking pushes the state around this ring to update the estimate of position. Yeah?
So there's thousands of neurons here, and you said you're sampling like 35?
That's right, that's right. I mean, different data sets have some plus or minus.
Raises the question of how many neurons are needed to encode the manifold.
Yeah.
And also, I guess, if you have thousands that are encoding the same thing, how is all that integrated? Here you're integrating it because you have a computer that gets to look at all of them. Are they in the actual circuit? You literally have like input from all 1,000 of them?
Yeah, that's a fantastic question. So there are models for how the circuit would work. So it's not just representation, it's a circuit that has to do some dynamics, which is has to take these angular velocity inputs and convert them into positional inputs. So the model is simple, I can describe it. So imagine taking a set of neurons, arrange them on a physical ring, and then allow every neuron to interact by exciting the neurons closest to it and inhibiting everybody else. So if every neuron does that in a rotation invariant way, then the connectivity is strong, then what happens is that, if by chance there's a linear instability. So basically, if you excite one neuron, then it'll excite all the neurons it's connected to in an excitatory way. There's a big activity bump locally, and everything else is silent. But because the connectivity is rotation invariant, it means that actually that fixed point, which is this one bump of activation, is also true for every possible rotation of that bump around the manifold. So now we've created a set of fixed points that lie on a ring, that it's a one-dimensional set of fixed points, and they're all related by the symmetry of the weights, which are this rotation invariance in the weight matrix. And so really, yeah, the idea is that somehow each neuron has some excitatory coupling with its local neighbors, and then maybe indirect inhibitory coupling with all the rest, at least statistically, yeah. And yes, and I mean the thing that really makes me wonder now, so until now the question, you could ask the question, why are thousands of neurons involved in encoding this one-dimensional variable, but you couldn't be sure it was really a one-dimensional variable they were coding. In fact, the typical situation in neural circuits is that neurons are multiplex the data that they encode multiple different variables at the same time. In this case, we can see the manifold, the whole manifold across all states is just one-dimensional. And so now we really can say it's one-dimensional and ask why. Okay, so yes?
So it's one-dimensional, but don't you have a different ring for every environment?
No, you don't. So it's the same in this system. So so I didn't mention it, but yes, you can put the animal in different environments, and the cells maintain their pairwise firing relationships relative to each other. It's the same neurons. They have the same offset in their tuning curves, which means that they're really on the same ring. And moreover, like the fact that in sleep you only see the same set of states. There's nothing special about the one environment, right? You see the same set of states in sleep, suggests that really that is the only set of states the system occupies. Because in sleep, there's no preferred environment. Yeah, great questions. Okay, so that was mammalian. That was a rodent. It was a rat running around. No, it was a mouse running around. This is actually, and actually in the mammals, we don't know the spatial organization of these cells within this area. It's not clear whether cells that have similar preferred head angles are co-localized, and others that are further apart are not. But the fly has a very interesting physical topography to its organization. So flies have also a head direction circuit. It is in this, there's a head direction circuit in these two areas, the protocerebral bridge and the ellipsoid body. And I think this is a movie, so let me try to play it. Oops. I'm not able to play this one, but I can just summarize it for you. So this is the protocerebral bridge, and it's actually, this is a calcium imaging, and these are like two bumps of activation. And then as the head direction of the animal moves, the bumps move. So here is a plot of the bumps moving around as the animal moves around. This is the ellipsoid body, similar physical bump of activity that you can see, and the bump moves in tandem with animal movements. So this is a physical ring structure in the fly that maps onto this abstract ring that we talked about in the state space of the encoding of these thousands of neurons. Now, I should point out that a physical ring structure is neither necessary nor sufficient for 1D encoding in the state space, because everything that I told you in the mammals, it depends on a certain connectivity in the model that I described between cells, but then you can keep the connectivity fixed and scramble the locations, and you would still have the one-dimensional ring dynamics. And similarly, if you have a physical 1D ring that you can see like this, again, to eliminate the possibility that the animal's encoding other variables, one would again have to look at the state space, right? So once again, there's some information that you need besides the physical layout of cells. Okay, so I want to describe much more briefly two other players in the spatial circuit. So now we're going to talk about piece two. So instead of a one-dimensional ring compass, we're going to talk about a two-dimensional Euclidean coordinate system in the brain. And so we've moved from the thalamus to the entorhinal cortex over here in the circuit. And this entorhinal cortex contains these remarkable cells called grid cells. So these are two grid cells that I'm showing you that are recorded from next to each other. And these cells have a very regular response to the spatial position of the animal. So this is the position X and position Y of an animal running around in a square environment. So we're looking at the environment from, for example, above. The gray is the physical trajectory of the animal as it runs around. And then the red dots are spikes emitted, the positions where the spikes were emitted by a single cell that's being recorded as a function of that position. Okay, and then this is another cell, same thing, same animal, simultaneously recorded. Okay, and I think that maybe on the right plot it's easier to see. You can see over here, these are also nice responses of the cell. So these cells are called grid cells for, I think, obvious reasons, because they have this remarkable grid-like periodic response to a non-periodic variable, which is the position of the animal in 2D space. Now, there's a lot of interesting questions, coding theoretic questions about why the brain would encode a non-periodic local variable with a periodic global signal like this. In fact, that's a question I'm very interested in. We have a bunch of work in that direction. I won't talk about that. I rather, again, want to talk about some discoveries about how this representation looks in the state space and whether it occupies some low-dimensional manifold structure. So, okay, so these grid cells, this is empirical results with data analysis. It turns out that grid cells have an intrinsically two-dimensional and invariant representation across environments and across behavioral states in the same way that those head direction cells had an invariant representation across behavioral states like sleep and waking on a 1D ring. So here grid cells maintain their pairwise relationships relative to each other in familiar and novel environments. In fact, even in 2D, even when you go from 2D environments to 1D environments along 1D tracks, then it turns out that the grid cell responses along the 1D tracks look like 1D slices through the canonical 2D response that they have. In fact, pairs of cells that you record simultaneously have parallel slices, as you would predict from these like 2D models. And moreover, the grid cells maintain their spatial tuning relationships relative to each other across waking, REM, and non-REM sleep, whereas these other cell types, place cells in the hippocampus, do not maintain their spatial relationships. So these are sort of like indirect evidence, right? You're showing that the pairwise relationships, the pairwise correlation matrix between these grid cells is preserved all the time, right? So this is, again, another form of invariance. In fact, similar models for grid cells, as for head direction cells, predict that, a circuit of grid cells that have, a common period of the spatial periodic response should lie in state space on a torus. Okay, and so the prediction is that the whole circuit of grid cells, again, thousands of neurons that have a common period, should just be a torus in the state space. So this is an analysis. This is an analysis based on synthetic data from the model, and in the synthetic data, you should see tori emerge rather than these 1D rings. And in fact, we're awaiting a sufficiently large data set with collaborators who are using these new neuropixels probes to directly validate this prediction by unsupervised manifold discovery in the same way as we did before. Okay, so one last fact on grid cells before we move to the third piece. The 2D response geometry of these grid cells and the prediction of their torus-like geometry of their response and topology of their response is actually embodied in the physical layout of cells in the mammalian cortex. Remember that in flies, the one-dimensional abstract state space ring is actually embodied in a physical 1D ring in the brain. So it turns out that in grid cells, the physical layout of the grid cells reflects their intrinsic, their abstract periodic response structure. So this is just showing, so this is beautiful. Oh, and I should mention the 1D grid cell work was again in collaboration with Sam, who's going to give the second talk, and with David Tank's lab, and this is all data from David Tank's lab and analysis from Sam and others from David's lab. And so this is neurons, different neurons recorded in the brain. So these are in spatial brain coordinates. They're colored according to the spatial phases of the response. So they have these periodic responses, so we can characterize them by a spatial phase. And then it turns out that they're, the relationship between their spatial layout and the spatial phase of their response to physical environments is related to what you see in their abstract responses to space. Okay, so the grid cell part went fast, but I think I just want to say that we have models, dynamical models for how the grid cell response can emerge, how grid cells can take velocity input and give real-time estimates of position based on those inputs, and those models predict that the grid cell response should lie on a torus in the state space, and then there's data, indirect data, that they do lie on this 2D torus, but we're awaiting more data to make a direct visualization that the data lie on a torus, but then there's also this physical architecture of grid cells that is consistent with their abstract representations. All right, so now these are all rigid, right? These are both representations that persist across environments from waking to sleep, and actually are quite stable over the lifetime of individuals. Although it's true that we don't have recordings over years where we can directly test that, but it's expected that these representations will be stable over lifetimes. And I should mention that grid and head direction cells have been found across mammalian species, from bats to mice to rats, and also some evidence in primates and humans. Okay, so I want to now come to the question of manifolds that are not as rigid. Okay, so the question is, where is the learning part? I described how in the SLAM problem, you have to have the ability to not just infer things like abstract 2D spatial coordinate and compass direction, but you also want to build maps of different environments and the objects within them, and specific layouts of different environments. So there's also a different area, the hippocampus now, that takes the input from entorhinal cortex and also has a source of input from another part of the entorhinal cortex that doesn't contain grid cells, but encodes things like objects and walls and other things in the environment that are conveying more information about the external sensory world than about the internal estimate of motion. And so these hippocampal cells are called place cells because in 2D environments, as animals just run around and densely explore two-dimensional space, different cells have spatially localized responses at one location and not others. Okay, so cell 1, cell 2, and cell 3. Okay, so recently it was shown, again from David Tank's lab, that these place cells can do other things besides encode spatial location in the physical world as they move through it. They can encode a much more abstract notion of location in abstract conceptual spaces. So this is an example of an animal that's pressing a lever, and when it presses a lever, there's an auditory tone whose pitch changes in some direction, in one direction. And so the pitch is increasing, and then there's a target zone. When the pitch enters some target zone, the animal should release the lever, and then it gets a reward. And the pitch can increase at different rates each time it presses the lever. So it really has to have, it can't just be encoding time and release at a fixed time. It has to wait for that pitch window to arrive and then release the lever. So that's the task. And when they record from place cells from the same animal, in this case, they find that the response of these cells is again locally peaked and modulated by position, now not in physical space, but in this auditory pitch space. Okay, so that's quite an interesting finding. And so Sam is going to talk in much more detail about some of the analyses of representations in these spaces. But I just want to mention two things before I stop. So the finding is that in the sound task, if you look at these, average responses, they look like they have these nice peaks. But if you look trial by trial, so this is trial by trial, there's quite a bit of variation in the responses, you can see. So this is the sound frequency space. This is each trial, the same cell is responding at different locations. And not only is the location shifting possibly in this space, but also the amplitude changes. So the question is, if you're really talking about these low-dimensional manifolds, where is this variability coming from? Right? Like, how do you explain so much variability in these traversals across, the same external space, and if the internal space is also very low dimensional? And so there's a couple of hypotheses. One hypothesis is that this variability is just like some high dimensional noise in the system, and that explains the variability. But another possibility is that there is this low dimensional encoding of this variable, like tone, right? This is a hypothesis slide. So there's like this tone that varies, in a systematic way, and you've got a manifold that represents that. But maybe there's some other low-dimensional structure in the manifold that exists, and it could be that, variable responses to the same measured variable consist of different trajectories through this low-dimensional space. So in other words, it's like saying that the noise is not some high-dimensional fluff, but some low-dimensional variability, right? And the question is that true? Like, is the noise high-dimensional? Is this variability high-dimensional or low-dimensional? And so so the prediction would be that if you look at different variations, they really are structured in some way. So it's possible to go from this location to this location in the sound space, but you always have to go through either this orange cell's response or the blue cell's response. It's not possible to go from purple to green states without going through at least one of these. But sometimes you might go through this, and sometimes you might go through that, right? That would be, a kind of low dimensionality. So then this is the last slide, and I'm going to stop here right after this one. So the finding is that if you plot this is the 2D spatial task. If you plot the responses, this is the manifold now projected to 2D. This is like place cells. I think it's about 50 place cells, right, Sam? And this is now taking the manifold, projecting it to two-dimensional space, and then color-coding the manifold according to the physical position of the animal in X versus Y along this 2D spatial task. You can see this systematic gradation of the response of the circuit to this two-dimensional task. But if you actually look at the manifold, the high variance directions are actually three. There are three of them. So it's not a two-dimensional space, it's a three-dimensional space. And in fact, the deviations in the single trials are quite consistent with just different traversals through this three-dimensional space. So it's a very low-dimensional noise. And the same is true in the sound task. So I think Sam is going to talk a lot more about that. So what I want to do now is I just want to end with my conclusions and say that the brain contains some very rigid and structured low-dimensional internal representations for these key latent variables that it extracts from the world, right? Compass direction, position are not supplied to the animal directly in any sense. It has to use all its senses to infer those variables, and it represents them using this very low-dimensional structures in the brain, right? And so, so we used unsupervised discovery methods to reveal this low-dimensional structure and find that there are these very abstract low-dimensional representations, and these form very explicit representations of these latent variables in the world, some representations are invariant across environments and states, and others are really based on the task, because the same neurons can encode this two-dimensional space with the three-dimensional manifold, and the same neurons can then do it for one-dimensional sound task using a two-dimensional manifold. So that's a reconfigurable example of a manifold. Thank you so much for your attention. And a lot of this work was done by Rishi, Burke, and Biraj from my group, with many collaborators, and especially Sam and David Tang from Princeton. Thank you very much.
Right, my name is Sam Lewallen, and I'm a postdoc at Harvard, like Liz mentioned, and my background is actually in pure math, but I've now been doing neuroscience. My PhD is in pure math, but I've been doing neuroscience for a couple years now, including lots of work with Ila and David Tang at Princeton, and so now I feel like I just don't know who I am. It's good. So, okay, so this is going to be pretty free-flowing, and depending on how far, I'm just going to kind of probably drop dead at the end, and that's totally great, and that's what I kind of intended. And if people are interested in various things that I bring up, which is mostly going to be elaborations and more details about things that Ila mentioned, then I'll be around today, and I'd love to talk to people here about lots of stuff, including brains and genetics, which I'm very interested in. Okay, so I'm going to have one or two slides just on this kind of basic idea about manifold learning and various ways that I think about it in general and in respect to the brain and in the context of data analysis. And then, depending on time, I'll talk a little bit about the details of the methods that Ila referred to in her head direction discussion, and that was joint work by Rishi and Ila. And then probably the bulk of what I'm talking about will be further elaboration of this project that's joint with Ryan Lowe, who's now a postdoc at UCL and was a graduate student at Princeton when we did this work together in David Tang's lab, having to do with manifold learning as applied to the hippocampal representations that people are now calling cognitive maps, which at the end of the day, to me, the most interesting thing of all this business is these amazing things we find in the brain, and that's what's so exciting about it. And actually, I think I even have a little comment here, right, about that. So before I get into the kind of more mathy stuff, which I'll start off with, it's going to be kind of this little overview, and then mathy, and then more overview. I just want to make this kind of put up this slide about our point of view. So Ila already had a slide like this, and I don't know, I'm not going to make the comparison directly, but one sort of simple-minded or sort of practical way to think about the thrust of this whole set of projects about manifold learning and neuroscience is that for a really long time, most famously associated with work by Hubel and Wiesel, where they, I'm not sure people here are familiar with it, but it's sort of iconic work in neuroscience where recorded from cells in visual cortex, and earlier it was done by many people in the retina of lots of animals, and you would find that the finite activity of certain cells correlated with very repeatable things in the world, kind of like what Ila was showing with the head direction cells. But in this case, in the visual cortex, it was sort of various visual features in certain parts of space that the animal was attending to or looking at, or at least was in front of its retina. And so that became, so that started this whole kind of industry of, you could record from single cells using electrodes, and then later on you could record from small numbers of cells, but still kind of minute numbers of cells, and you would painstakingly sort of correlate the behavior of various cells with various stimuli in the environment, and that became the entire industry. And sort of, I would say most of neuroscience still does a lot of things like that. You sort of record cells, you see what the animal is doing, and you see how they correlate. You do a correlation. And I guess one question is, how far can that take you? I mean, at the end of the day, these cells aren't just kind of representing the world. They're doing some kind of computation, or maybe we don't even have the language yet for what they're doing. And so although we don't know exactly where to go, we kind of hope that tools like this kind of intrinsic approach where you think about the entire population of cells as having some sort of structure to their activity, can kind of give us a new language almost to talk about neural computations or things like that. So sort of the whole thrust of this, a funny way to put it is that when you still see tons of presentations where this is going to be kind of a little negative, but where you see just sort of just a list of cells and here's their correlates, right? And you just kind of say, is there anything else we can learn from the system besides that? Is there any other way we can think about it? So it's sort of just providing language. I don't think anyone really knows where to go, but we hope that one of these things will end up being really insightful. And again, the technology point, which is important to realize, is that we can, we're having this huge growth in the number of cells we can record. People are probably aware of things like revolutions in imaging technology, calcium imaging, and things like that we can now, we probably couldn't do analyses like this before. Okay, so that's an overview comment, and I had this little comment here about this. So I would say that both the work that I'm going to talk about and that Ila presented in terms of what we're providing algorithmically and conceptually is sort of ways to sort of get simple properties that are sort of geometric or have to do with the manifold structure of these activity population, these subspaces of activity space, and kind of just using information about that to reflect, to learn something about the system. But like sort of when we talk about manifold, we're having a picture like this. It's still a very simple object, I would say, right? And a sort of obvious question with Ila's presentation, even in terms of this head direction circuit, is what if you're, that seemed almost too simple, right? Like, what is the brain actually doing? It can't be that it's a bunch of these little rings. There must be some more structure there. And how would you talk about that structure geometrically? Is there a geometric language for talking about computation? And so, I think of understanding what the brain is doing is sort of this big frontier where we don't really have a language for talking about that. And I just think it's a really interesting kind of synergy with things like in, genetic programs and stuff where we don't really know sort of how to say how a set of genes turning on each other on and off sort of goes into making a limb or something like that. And sort of, do we have the right mathematical language? Is there maybe some notion of like a geometric framework for computation? Anyway, so that could be where this kind of thing could go, but I have nothing else to say about that. But right now we're just sort of talking about simple manifolds. Okay, so and also if anyone has any questions, please let me know. Okay, great. So, oh shoot, this is kind of hard to read. So in these next two slides, I just want to kind of talk about what I think of as what I mean by manifold learning, just so we can all be on the same page. And right, it's sort of this funny distinction between manifold learning and dimensionality reduction, which is an interesting thing that we could talk about too. I mean, I think Ila and I both agree that just linguistically almost and kind of aesthetically, dimensionality reduction feels sort of like we're just cleaning up the data or something so we can use it for something else. Whereas we want to understand, is there some intrinsic structure sort of intrinsic to the generative process of the data, for example, that really sort of tells you something about the system, sort of more of a scientific point of view. And that's a kind of not really a true distinction, but okay, so just wanted to warm up with this PCA example. So PCA is sort of like this kind of simplest, linear manifold reduction technique, which I'm sure everyone here is familiar with, although it's not always presented this way, right? So just I'm going through what's the setup. The setup is that we have k data points, x1 to xk, in an n-dimensional space. So like in our examples, it'll be n neurons. And the goal is just to approximate the data with a lower dimensional linear subspace. And the key point is that we're going to have force it to be linear. And let's say little d is the dimension of the subspace. So in this picture that I spent all of yesterday doing and therefore didn't prepare the rest of the talk, so I hope you guys like it. That's only partly a joke. This is the d equals 2, little d equals 2, and big N equals 3. Right, great. And the data points, I'm representing them by these white dots here. And then, you can say, so sort of how would I want to, I could have chosen these to look more two-dimensional. We're going to try to approximate it by a two-dimensional plane, and we need to have some notion, there is no two-dimensional plane that goes through all these points, so you want to have some notion of what's a good approximation, right? And there's lots of things you can imagine doing, and that's an interesting point. But one kind of natural thing you might do is you might say, well, look, I'm going to take this plane, and for every point, I'm going to project it orthogonally onto the plane. And I'm going to measure the distance from that point to the plane, okay? And call that this E of xi, depends on the point I'm projecting and the plane I'm projecting to. And then just take the average, and in this case, it turns out to agree with these least square type algorithms. You want to take the square. You could not take the square, right? And you can do a lot of different things. But in this case, if you can define a total error for a fixed single plane to be the average over all the data points of the squared distance from the point to the plane. Great, and then, so as I wrote here, the algorithm is going to choose the good subspace to be the one that minimizes this error, okay? The average squared distance of all the points from this plane. And so it turns out that algorithm gives you, it's basically the same as PCA. Okay, so remember, we as input chose this d equals 2. So in general, d can be any number smaller than the total number of dimensions in the original space. And so in particular, you can see that if you just are, the comment related to one of the more traditional ways PCA is described is that if you take your data vectors, which are n-dimensional vectors, and line them up into a n by k matrix, right, which I'm calling X here, then if you let C equals XX transpose, it's the covariance matrix of the data, and then the comment relating PCA to this plane is that the plane L that has this minimal property, which is the best plane, is spanned by the first D largest eigenvectors of the covariance matrix, right? Where the size, I'm saying, measuring by how big the eigenvalues are. Okay, so PCA is the best fit plane. Okay, so that's sort of linear dimensionality reduction, or linear manifold learning. And so what Ila and I are going to, are talking about is just doing this idea of doing nonlinear manifold learning, right? So the idea is, what if the points that you're talking about lie in some high-dimensional space, but they still form a lower-dimensional structure, but that structure is not some linear subspace? Then what kind of techniques are available? And I don't actually know as much about the history of this in terms of data analysis and machine learning, I mean, as I would be interested to learn, but starting around 2000, which is when isomap and LLE and a bunch of other algorithms were published, there's been a huge amount of work in methods for doing this kind of thing. So I guess I was also going to go through the isomap algorithm. So I made a comment here. This is, hopefully I'll be able to come back to this later on when I come to our method that Ryan and I worked on. But there's a kind of a hidden thing here, which, depending on how math you are, you might think this way or not, that what did we use here to define the quality of this plane approximation? Well, we took the average distance of the points to the plane, right? And that distance, where did that come from? That distance came from the Euclidean structure of the space of, in this case, the data space, basically. Right? Don't forget that there's this funny thing that goes on that when you hand me a bunch of numbers in coordinates, that sort of feels like it's a vector. I can take its dot product with other things. But that vector structure may or may not be related to the real data, right? I mean, it's determined. This comes up a lot when people talk about, I want to correlate things, and how do I choose the scale to match them? Even, right? That's a very simple question. That, if I want to talk about things geometrically, I have two axes. If I just double one, it goes from being a sphere to being like an ellipse or something. And that is just a choice of unit, right? So more generally, if you sort of unpack that, the metric here, there's a kind of geometric structure, but it's coming from something to do with the way that we're representing these neural activities as real numbers. Anyway, so that's food for thought. I mean, in general, the way a lot of these manifold learning techniques work is that they, where the kind of creativity or conceptual interest in an algorithm might be to choose an interesting metric that's different than the one you might naturally think of. And that's sort of what Ryan and I, one of the things that we worked on, which I hope I can come to. In our case, the metric had to incorporate dynamics of the activity, the kind of fact that it's a time series, which Ila alluded to. Great. So I think in the interest of time, I won't go through this in the same detail I did through the PCA thing, which is maybe the wrong order to have done it in. But I just want to really quickly sort of give the general idea of what isomap is doing, okay? And as far as concepts go, I think it's very much represents what Ila and I are talking about in terms of nonlinear manifold learning. Right. So again, the basic picture to have in mind is that we have a bunch of data points here. I drew them rather as trajectories. And we have these three axes, neuron 1, 2, and 3. And but basically some kind of structure or correlations in the activity results of the fact that the trajectories are not filling up this whole space, but relying on some, in this case, sort of two-dimensional or ribbon-like spiral manifold. And in the previous discussion, we were talking about distances on this manifold that came from this ambient space. But the idea in isomap is to try to kind of capture the notion that there should be a notion of distance where it's still defined by saying how long does it take to get between 2 points. That's their distance. What's the shortest way to go? The length of the shortest way to go. But now I force myself to stay on the manifold, right? That's what we mean by an intrinsic distance. It sort of says, so intuitively the distance between this point and this point, between this point and this point would not be the distance along this line, but it would be the distance traversing the entire ribbon, like that, right? And so isomap is a very elegant, sort of simple way to capture that kind of idea, and it works like this. You take this is the same bunch of data points, x1 to xk in Rn. And again, the goal is to approximate the data by this now nonlinear subspace. And we're going to first define this new distance metric called the intrinsic distance, okay? And here's how we're going to do it. It's very simple. We're going to imagine, you need to think of a graph or a matrix whose ijth entry is just the distance, the Euclidean distance from point xi to point xj, okay? And now we're going to throw away, so you imagine those connected by a straight line. But now we're going to, and that distance is the length along the straight line, right, from xi to xj. And so we're going to fix a parameter, which is a little number k, which intuitively says, if I'm a point here, what are my neighbors? You know, k is going to be the number such that we call the neighbors of this point the k closest points to it, okay? So we're imagining that we're taking a point in our data set xi, maybe it's here, and we have this matrix of distances, right? And we're going to keep the k closest distances, but we're going to throw away all the others. We're not going to allow us to traverse those paths, right? And now the idea is you can do that with respect to every point, and you have a new set of paths, right? It's a subset of the original paths, right? But it's cool. You have to, algorithmically, there's a whole field of sort of shortest paths on graphs. It's like an entire sort of algorithmic problem. And right, the idea is that if you just think of it, if you're an ant walking along these paths, these little short paths, you can make a long path, but it's a collection of little paths, right? And so the distance between 2 points is now going to be the shortest distance between the length of the shortest path, where I'm forced to go not in general through this whole big space, but along these k nearest neighbor paths. Anyway, so just that was a bit fast, but just the hope is to get some kind of intuitive notion. But this is basically what you've done is you've replaced this original distance between your points by a new distance, which I just wrote this d iso xi xj. Okay. And then that's the first step. And then the second step is a very generic step in a lot of algorithms, including the one I'm going to talk about, which is, so step one was that we replaced our original kind of extrinsic metric by a new intrinsic metric. And then the next step is we want to embed this in a, let's say, two-dimensional space in this case. And what we're going to do is we're going to call yi and yj the image of xi and xj in this two-dimensional space. And we're just going to solve an optimization problem, okay, which says that I want the distances between pairs of points in this embedded space to be as close as possible to the distances that I constructed, the true distances, so to speak. And this is called multidimensional scaling in general. And you guys may be familiar with something called classical multidimensional scaling, which is not what I described here. That's a kind of more linear algebra style algorithm, but this is actually simpler to state. It's just a very sort of generic optimization algorithm. Embed the points so the distances in that embedded space match the distances I have here. And you can see from that point of view the power that you get in choosing these distances. It's sort of like that's capturing the meaning of your algorithm, basically. So any questions about all of that? Yeah?
How do you choose the dimensionality of the embedded space?
That's a great question. Yeah, so you need to have some kind of goal at the very least to start with to make that choice, I would say. And there's going to be different paradigms depending on what you want. That's actually going to be a big part of what I'm talking about. And I would say in general, I'm not completely satisfied by sort of the set of the options that I've explored. But I mean, so for example, if you were if you set yourself up so that you can kind of predict data, then you can do a sort of cross-validated type thing from your intrinsic manifold. That's one approach. But there's subtleties there. So in general, I think Ila kind of, it's not unrelated to this question that Ila kept alluding to, that she didn't want to guarantee that there isn't structure in this kind of space around the ring that she was talking about. And in general, I don't think we have, we don't want to claim that certain things are noise, but we hope to be able to find some structure. Okay, great. So, right, so the next two parts I'll talk about, so that was the kind of overview of what we mean by manifold learning. And so that's a general approach, that we're going to try to capture this intrinsic geometry of these activity vectors living in some high-dimensional space. And so I would say that what Ila and I worked on is to sort of contribute, sort of go one next step in two different directions and look for additional structure, or kind of take it further, things that you can talk about in the data from this geometric point of view. So one of the things that was kind of hidden in what I was talking about here was that when I'm doing this whole embedding procedure, I'm requiring yi and yj to live in, I'm taking these high-dimensional points and I'm trying to find yi's and yj's that represent them well. And by definition, I'm having these be, in this case, two-dimensional vectors, right? So what that amounts to is that I'm asking for a mapping from this high-dimensional space into a two-dimensional Euclidean space. And as Ila pointed out, the circle is not a one-dimensional Euclidean space or a two-dimensional Euclidean space. It has some other topology. And I would say that despite kind of tons of work on manifold learning, there's a very small number of methods that just account for that. And I don't know, I guess you can ask how general these kind of topologically interesting manifolds are, but certainly loops is a very common type of thing that you can imagine coming up in a lot of brain circuits and computations and things like that. And so sort of one of the things that Ila and Rishi contributed was to kind of develop a framework, as she alluded to, for intrinsically, so you can think of this green curve is a bit like this. It's a 1D space. It has a variable called theta, as she alluded to, or maybe she called it alpha, I can't remember. But that represented this sort of circular variable around the manifold. And that's kind of plays the equivalent role to this 2D thing that I was embedding into here, right? So you can imagine if yi could just be in some generic space that you could specify, then specifying the loop would give you back sort of Ila's algorithm, basically. That's almost literally true in terms of the algorithm that Rishi and Ila used. The only difference is that instead of letting the points, let's see, yeah, I guess you have to construct the space first. That's the main difference, right? And I think I probably won't go through the details of that. Yeah, maybe if I had time at the end, I'll actually describe the specific algorithm that they use. Yeah.
Sam, maybe one thing that might be helpful to say is that there's a distinction between like local dimensionality reduction and then global dimensionality reduction. So you can think about, if you take a manifold and you can unfold it into a Euclidean space that's lower dimensional, then it's like a global embedding, right? You've taken the whole manifold and the whole thing can be embedded globally in this lower dimensional manifold. Here, and hopefully that low dimension equals the intrinsic dimensionality of the manifold. And in this case, you can think about it, if you're thinking about it as a Euclidean embedding, you could say it's locally Euclidean, but globally not. So you can think about it as a local, so you could imagine just sort of creeping along the manifold and then doing a local dimensionality reduction on that in a Euclidean sense, but then globally it's not Euclidean at all.
Right.
So it's like local versus global, maybe that's how I...
Right, and even in particular on this slide, sort of what Ila's alluding to is that, it's funny, you can ask even what sort of is there beyond manifolds, and we've been using this word manifold without really defining it or anything. But one way to think about how it's defined is that what it means is that locally you are one of these Euclidean spaces that I was talking about. And so that locally you don't need to worry about this ring type thing, and you can just, if you're, locally you're determined by one number, which is your dimension. And maybe in this case you'd have dimension 2, and in that case you'd fit into a two-dimensional thing. But then this ring thing, it's very kind of fundamental that the ring property is intrinsically what Ila was referring to as a global property. It's the thing that you get by piecing together lots of these local things. So so this issue about needing to do a parameterization that's not sort of the standard one only comes up when you're referring to kind of global properties. And, there's again, everything about sort of beyond vanilla manifold learning, sort of how can we explore these neural computations. This idea of everything as a manifold, and that that's also probably false. I mean, a simple thing that manifolds have to satisfy is that they are a single dimension throughout if they're connected, right? And presumably, whatever dimension of a computation means it's sort of fluctuating and changing, and things aren't quite continuous. And what does a computation look like geometrically? So I think there's a lot of, it would be cool to have a language like that where I could almost, sort of search for things like motifs, geometric motifs in neural data that's more complicated than this stuff. But, okay. So, I think for interest of time and to get to the kind of mind story, I won't go through the details of this slide, but the thing that I can talk about that I was interested in is sort of the detail later on or afterwards is the details of this topological data analysis algorithm, because it's sort of, in terms of the things I'm saying, in terms of finding new ideas, it does represent a kind of different sort of way to characterize properties of the data, of the manifold geometry, or topology in this case. And an interesting thing is that algorithmically it uses the same kind of neighbor graph in some sense as the isomap did. So what you do in this persistent homology stuff, which is very cool, is that you we had this parameter k in our previous discussion, which had to do with sort of what counts as local on the manifold, right? You imagine in your head visually that you have a sort of ball around every point on your spiral that represents sort of what counts as being local on this intrinsic manifold. And you can imagine, as Ila was describing here, to vary that k and to get kind of coarser and coarser approximations of your manifold and sort of see how things change as you vary that parameter. So that's sort of and you can hope to kind of A, have a more robust algorithm, but also B, you can hope to, this sort of did come up in this discussion from Ila, but again, in this sort of theme of more complicated structures, you can imagine that there's some kind of really multi-scale structure in these neural computations. And we really want methods to uncover that, which probably I think everyone in data analysis wants, sort of automatic methods for multi-scale structure discovery would be great. Okay, so I think, so that's sort of end of that talk. Now just another talk. Okay, no, so what I want to, actually, no, sorry, that's wrong. Take it back. There's one sort of final algorithmic aspect, which I want to allude to. So this is a kind of general slide. Now I'm talking about joint work with Ryan Lowe. And this is a overview slide of the method that we've discussed, that we've constructed, which is called, this is Ryan's acronym, it's pretty great, Manifold Inference from Neural Dynamics, so MIND for short. And I'm not going to have it, it's got a lot of details, and one comment I was going to make again, sort of in this philosophizing algorithmically, there's a certain notion in which PCA is kind of unique in some sense. It's really kind of a canonical algorithm. I mean, that's not, there's other kinds of linear dimensionality reduction algorithms, but there's probably, you can define a notion in which that's true. But I don't think there's any hope to have a kind of canonical nonlinear dimensionality reduction algorithm, right? I mean, it's sort of like, who was it? I think Helmholtz, maybe John knows someone else, it was attributed with saying that linear versus nonlinear is saying like banana versus non-banana or something. Yeah, like, it's just, yeah, I mean, you can always create categories, but they should be more useful than that. But, so, this came up I was at a little mini conference. Ila was there too with a bunch of, manifold learning in neural analysis. And, so I think one experimentalist who I love, he's one of my friends, he was complaining that, you guys have to stop designing algorithms and just choose one already and like fix that on it. And I don't think that's the right attitude, although I appreciate what he was saying. I mean, I think that there isn't enough organization among theorists either, but I think that's one comment about that. Yeah, I don't know why I thought of that. Sorry. Right. Okay, right, because the algorithm is complicated, so I'm being defensive. Okay, but the one thing, so I don't have to go through all the details. A lot of it is sort of things that I would, but that being said, I think given that I have a pure math background, there is a difference of culture. I think that algorithmically, I don't know if people here will relate to this or appreciate this, but I think there is the possibility of sort of talking about algorithms in a way that emphasizes more the structural similarities between different algorithms. I feel like that's not done very commonly in machine learning in my limited experience, and it's frustrating. And so for example, we are, a ton of this stuff is about how to choose the size of your neighborhood in your algorithm, in your manifold learning thing, and we have some approach that we like, but I don't think it's particularly principled, and I would be happy to change it. But it's kind of fascinating it's Ryan's idea of using these kind of random forests, they're called. But I'm not going to touch on that. But the one thing I want to talk about just conceptually, because I think people here will appreciate this, is this notion of what we're doing to define the distance. So this came up in discussion in Ila's talk after a question, I think. Ila, as we discussed, I guess I should use the board for something. This may be a bit, right. She was talking about, as we discussed before, this intrinsic parameterization, which we were describing with the isomap algorithm just now. And so there's a notion of distance here. Oh, is that, okay, it's better. Thanks. Oh yeah, shoot. Can't see that, it's okay. Right, so this is Ila's ring manifold, or the rat's ring manifold. Yeah, you probably haven't, yeah, I mean, it's amazing, yeah, there's a lot of evidence that humans have these same representations, which is pretty amazing. Anyway, right, so, this came up in terms of a question, but in Ila's talk, but, what this isometric algorithm does is lets you define this intrinsic geometry rather than the extrinsic one. But it's still coming from the local Euclidean geometry of these neural data vectors, And so, for example, you can ask, I mean, here's a stupid thing. If you square the vectors, how does this change this, right? I mean, we get, why do you even get a vector? Well, we have this whole issue in neuroscience of, do you want to think about spike times or do you want to think about firing rates, right? And what Ila referred to sort of once or twice probably was that she binned and not she, but everyone, we binned, and I'm doing it here too later on, binned the firing rates or smooth them to create these, bin the spikes that you get from the electrodes to actually create kind of real valued things, right? And now we're getting a lot of data from calcium imaging, which is a totally different signal. And it's already sort of continuous in some sense. It has to do with the amount of fluorescence that a camera basically picks up coming out of cells. And there you can say, there's some relationship between that signal and the original and the spikes, but or the smooth firing rate you get out of the spikes. But you'd be worried about what that relationship is, right? And how does it influence your algorithm? And does your algorithm, is it really just picking up something that's an artifact of that signal, property? And so, Ila, at the end of the day, she was talking about topological properties, like having this ring, and that probably will be invariant under various changes in, imaging modality and stuff. But you could worry that the metric properties of your manifold, like the actual length of these various lines, wouldn't be. And also, it's another interesting point that Ila alluded to, that she saw sort of the way the angle in the head direction coding related to this metric was sort of homogeneous across the head direction circuit, right? And that actually, although it involves the metric geometry, is a property that's sort of independent of it in that it's a symmetry property. So that's another nice observation, that it's also kind of a thing that could be true in many different geometries. But the thing that Ryan and I worked on was a different way to kind of deal with this way of what's a more intrinsic way to define the local geometry. So I just want to touch on that really fast, and then I'll give the main point about our project, and then I'll probably be finished. So basically, the reason why, what we did, and the reason why it has this word, this MIND acronym has the word dynamics in it, is we're going to define a new metric that takes advantage of the fact that we have this extra structure that we haven't really used yet, which is that we just don't have a bunch of disparate data points, x1 to xk, but we actually have a time series, right? We're recording these spikes from this animal as it's moving around, and they come in a temporal ordering, right? So in particular, you can speak, instead of points, you can speak about trajectories on your manifold or in your high-dimensional neural space. And so what we're going to do is, instead of talking about getting a little, characterizing the shape of a little ball in our intrinsic manifold, we're going to talk about characterizing the transition probabilities in a little neighborhood of our manifold. So I want to basically, just conceptually, I want to define a probabilistic object, which is going to be the following thing, that if I'm here in this point in my intrinsic manifold, how likely am I to go in one time step to any other point on the manifold? And likely means empirically in the data, how likely am I to do that? And so just abstractly, you can define a transition probability, which is sort of probability to get from one point to another point in one time step. And you can imagine, sort of in a probabilistic framework, now estimating that from the data, right? And there's various ways to do that, which we define one. But then you get kind of a pairwise matrix probability of transitioning from one point to the other. And all I wanted to say is that we're then going to use that to define distances. And I'm just going to flash this up really fast, but there's a kind of a canonical way you can go from a transition style matrix. So if you're familiar with Markov chains, you can think of this like as a transition matrix for a Markov chain, except that now the variable is not a discrete set of nodes, but it's a continuous variable, right? So it's a process of some kind. And we just want to say that, this basically says that your second order statistics should look like diffusion with respect to this metric. And that's a way to define a metric. So if that was meaningful to anyone, then that's great. And if not, no worries. But the point is, we're not actually, so this is kind of interesting. And another genre of algorithms in neuroscience, which are very interesting, is that you actually model the dynamics with a dynamical model. And so what Ryan and I are working on here is something that I would say is a little bit in between. We're not saying that we're going to define a sort of geometrical structure on this neural activity in such a way, we're not saying that the neural activity is diffusing through it, but this geometry, but we're saying that the distances are such that if it were diffusing, the second order statistics would be, if the distances are such that if it were diffusion, what's the way to say this? the distances are such that the second order statistics of the actual dynamics are consistent with diffusion through that geometry. So it's sort of like intuitively, if you go really fast through a bunch of states, then they should be closer, even if it's the same amount of distance in Ila's sense, and conversely. Yeah?
The fundamental solution will look less and less like a classic Gaussian if there's other structure and you allow time to grow larger, right? This would be true for...
And even locally, it could not look like that if there's... And that's a, we haven't really explored this, but a great question, now a fun thing to do would be to actually say, at different points of my... Right, so exactly. So this model would actually be, you imagine in your head that you have this little ball in your manifold, and you have trajectories actually running through it. And you can now be able to ask the actual empirical distribution of trajectories running through a point, does it match uniform Gaussian around that point, right? And that may vary across the manifold. That should tell you something about how the system is working or something.
Yeah. And the local dimension.
The local dimension also, yeah, exactly. So, we hope this will provide eventually want to provide a framework for maybe asking about how different dynamical aspects vary across the manifold.
And how does this relate to diffusion maps, the Koiffman at all?
Yeah, so there's actually a lot of similarity with diffusion maps. It's actually not the same thing, it turns out. So it's funny, Koiffman et al., he's got a lot of papers, but as far as I know, in the original diffusion maps, they didn't actually do, this is almost simpler than what they did. They have this kind of diffusion distance, which is some kind of smoothed out version of this. Yeah. And that was important to them to get the smoothed out thing. But this is sort of the simpler thing that you would do that says, I want to actually match the geometry, not get a smoothed out version by averaging over paths or something. So, but there's a pretty close relationship.
I guess you somehow care about sampling rate here. Data points. You can imagine if you sample slowly relative in the head direction example, if you sample slowly relative to how quickly you're turning your head or something, then the neighboring points in time will actually be very far apart in angle.
Right, so that's true definitely. And then furthermore, you could ask another, you could imagine that being true in a way which is homogeneous across the manifold. And then you still have to worry about that, but that's probably still consistent with the way we're thinking about this. But in really case, it would be pretty different from the way the system would break down even more if you really, the structure of the system had very different dynamical scale, temporal scales at various points. And you couldn't even make, somehow in the background, if you would formulate it statistically, we're making some assumption about this time step makes sense to look at across the whole manifold, right? And that could break down too, which would be fun to also explore. But yeah, there's another question.
Yeah, just are they transition or BP are truly kind of invariant?
Well, that's another kind of similar answer. Yeah, definitely not. You mean like time invariant? Do you mean like stationary or something? That's a huge, that's like this kind of hierarchy, dream of having hierarchical analyses, dream of having non-stationary assuming analyses, and would be awesome, yeah.
But related to the sampling point too, right? This is not autonomous dynamics on the manifold of the system. Like I could imagine that if you did this during the animal sleeping, then it's some kind of autonomous dynamics, and then you're doing, it's like, right? It's really the internal dynamics of the system that are driving the states. But here it's like whatever the animal chooses to do. So it's sort of the visits of the different states in time and the spacing between them depends on the animal's behavior.
Yeah, that's right. It's which also depends on the brain. But no, I agree.
But it depends on what you're making it do, right?
Right. No, we aren't at all distinguishing between those things. Yeah, you can imagine some system for trying to understand that. Right. And we're not even really modeling the dynamics.
You could imagine starting the animal off here and then having it run really fast to here, and spend a bunch of time here, and then runs really fast to there. So then it's spending maybe it's slow in some regions and it's running really fast in other regions.
So it's kind of related to this question. Yeah. But then another version of that, which I don't know, Ila's aware of this, but it's kind of this amazing. It's funny. Again, we were at this meeting where there's sort of two communities. I would say no one really phrased it this way, but there are the motor people and then these kind of hippocampal people. And it's really fascinating, right? Because it's interesting how to think of the motor system. I also have no idea, but a lot of the motor tasks people are looking at is a very highly trained motor reach task. Let's say they're recording from the motor, part of the motor cortex of, let's say, a macaque monkey, in a very brief window when it's like reaching for something in a stereotyped reach. And there it looks like the dynamics only go in a single sort of straight line every time through a certain part of the space, and maybe when it reaches in a different direction, it goes on a different straight line. And so they have this picture that's very dynamical. It's right. So there's a fixed dynamical system that you're at one point and you go to the next point. Whereas, kind of related to something that Ila just alluded to, in this whole head directions picture, the the circuit itself is not determining where you can go on it. It's just constraining, sorry, it's not saying you can go to only one place given where you are on this ring that Ila was talking about, for example. It's saying you have a set of limited trajectories, but within that you can actually go everywhere. It's almost the extreme opposite. It's sort of like it's sort of a memory circuit that's waiting to be pushed along, it's sort of to be integrated through, but itself doesn't tell you where to go next. And I just find that cool. I mean, there's two parts. People in those fields use really different language. And which one is sort of a more canonical, a better way to think about brains? Maybe it's both. How do those different kinds of dynamical systems get embedded in neural circuits? Quick question? Okay, yeah. So let's see. I think in the last 5 minutes, I'll just give sort of an overview. And we have a paper in bioRxiv for all this work, which is definitely cooler than everything else that I just talked about. In particular, the coolest thing is this task. So maybe I'll just tell you what this task was. So right, I'm not going to tell you about the variability style result at all, because this task itself is almost kind of, or the finding that's represented by this, which Ila didn't get really a chance to touch on, so I'll mention that. So I'll just tell you, I'll just describe for this last part of the, I'll just describe what's on this slide. So this represents work from Dmitry Aronov, who was a postdoc with David Tenk when we were doing this project, and now he's an assistant professor at Columbia. So what Dmitry and David did was they trained these rats to do two different tasks, okay? And so the tasks are depicted here. So what's depicted here schematically is what people call this random foraging task. So what you do in a random foraging task is that you put a rat in a box, and then you, the experimenter, throws chocolate pellets, it's always chocolate pellets, in the box, so the rats are always high on chocolate, that's sugar highs. But and you throw them into the box, and the rat runs around, and you do that to try to get it to move around the box, and you can record neural activity while you do that. Okay. So it's kind of a funny task, but that's very standard. And then people, Ila alluded to this. People now, when you're recording from the hippocampus of this rat, you'll get these so-called place cells. So what I'm depicting here, this is a sort of classic plot of a place cell. What I'm depicting here is this 2D space here represents the space of the box, and each of these pictures corresponds to a different cell that's being simultaneously recorded from this rat's brain. And the coloration here, when it's higher, it means that you saw more spikes there, and when it's lower, these blues represent basically no spikes. Okay, and this is a 20-minute recording session. And so what you can see from these plots is that the activities of certain cells are localized to certain regions. That's what you can see from this plot. A more striking thing would be to point out that when you do, let's say, the same plot for the first 10 minutes of the session, the same plot for the second 10 minutes, it's exactly in the same spot. So it's really kind of a consistency to the place that the cell fires. And the really striking sort of memory-related aspect is that when you put the animal back in this box a week later, the same cell fires in the same place. So the basic picture that, this work was recently awarded the Nobel Prize in 2014, with, this activity in rats was discovered 10 to 15 years. It was discovered 10 to 15 years, let's see, 20 years, okay, some amount of time after this sort of HM story that Ila was alluding to, where this person, when he got his hippocampi removed for an epileptical surgery to treat his epilepsy, he lost his short-term memory, right, ability to make new memories. And now you're recording these spatial cells in the homolog, the exact same region in a mouse or a rat. So sort of it led to all these great ideas about, these interesting questions about why are you mapping space in a rat in a place where in humans has to do with memory, and with rats, is it just a general memory system? And we have these ideas about, going back to sort of memory palaces where you put things in different places and it helps you remember things. So is that somehow related to this activity? And so it's a really great, interesting field. But for a long time, there was this question that people would argue about whether, even in the rat or the mouse, whether this brain area, the hippocampus, was responsible just for some kind of spatial mapping or whether it was a general memory system. And we still don't know, but what Dmitry and Dave did was they said, I'm going to at least explore this in an abstract navigation task. And so they designed the following task, what they call the sound manipulation task. So here's how the task works. You're a rat, and you stick your head in a nose poke so you can't move around. They did that to try to get rid of confounding spatial variables. And then the rat is trained to push down on this lever, okay, in the behavior box. And when it pushes on the lever, there's a sound that comes out of the speaker, and in proportion to how much it's pushing on the lever, the sound increases in frequency. It always starts at 2 hertz and it increases in sound to 22 hertz ultimately. And the animal gets a reward if it releases the lever sometime in between this 15 to 22 hertz period. Otherwise, it gets a timeout. And over time, the rats learn to do very well. I don't have a picture of the behavior here, but the rats learn to often get the reward. And furthermore, Dimitri and Dave, so there's the actual speed and time through this sound sweep is related, like I said, to the force on the lever, but it's related by a gain constant. And that constant gets varied randomly from trial to trial. So what that means in detail is that getting to 15 hertz doesn't take the same amount of time on every trial, right? So that's sort of try to disambiguate time counting and frequency counting. Anyway, the long story short is what they find is that just like you found a spatial map in the hippocampus, you find a frequency map in this frequency task. Okay, and so the basic conjecture would be that you would always find these sort of maps of these kind of continuous spaces that your hippocampus must ultimately create as you're going through your life. And so that you can see it's very appealing to use the system and try to sort of talk about these things from this intrinsic geometrical point of view and sort of basically ask if they have properties that we're not seeing by just averaging over these, by correlating with a particular variable. So in the project that Ryan and I worked on, we argue that, as you alluded to, that in both these cases, you see these maps, but they're actually higher dimensional than these averages would make you think. And that's the work that I didn't get a chance to talk about, but you should see our paper in the bioarchive if you're interested. And that's the end of the talk. Thanks very much.